And we're live. Greetings, everyone. Welcome to the Evidence Reasons Academy. I'm rejoined here by Katie. We
Hello.
will be. Oh, hello.
Hi.
Hi. So we are going to look at the UNIPROT uh, information base, knowledge base, and I'll also be spooling Katie up on some of the specific proteins that I think would be good for her to study. And so study we will. So with that, my favorite protein, it just happens to be the one that I work on, but it is probably one of the coolest ones just to share with anyone, is the topoisomerase protein. There are actually very many families of topoisomerases in humans. There are at least there's topoisomerase 2 alpha and topoisomerase 2 beta. They are targets of chemotherapy because if you disable a topoisomerase, uh, if you disable both alpha and beta, you're dead. And so the chemotherapies are poisons that poison the action of topoisomerases, and so it kills cancer cells. Unfortunately, it's also very powerful at killing normal cells. That's why chemotherapy is sort of the last resort for treatment of cancer. You try to not give as you try to do some surgery and then chemo to get the residuals. Because it's very painful. It kills a lot of healthy cells. And so it's really, when they say it's tough medicine, it really is tough medicine. It's not really the first thing that we would want to be injecting people with. So because of that, because of the action of chemotherapies on topoisomerases, it has been an intense field of research, because you can also, if we can figure out how to kill selected cells or even certain bacteria or whatever, it's going to be a very powerful way to just get things, living things that we don't want in our body. So it's been an intensely studied enzyme. Enzymes, for those not familiar, is a biocatalyst that enables chemical reactions. And I'm gonna show Katie here a video, one of my favorite videos about the action of topoisomerases, and we'll talk about it a little bit. We'll then go to the UNIPROT website and start learning about the mechanics, the very details, and that will enable us to start doing preliminary research and start publishing. There's so much. We're in a situation where there is so much knowledge. We have these automatic sequencers and all these robots, these robotic machines that do a lot of the laboratory work for us. And we have actually far more data than we have time to analyze. And so these machines gather data and we start analyzing it and trying to connect dots and figure things out, figure out what it all means. It reminds me of archaeologists who go and just dig up as much as they can and then file it and catalog it. And then for the next 40 years, they're studying what artifacts that they've discovered. They're trying to interpret the hieroglyphics or piece together puzzles. This is the state of biology now. There is so much information. We've not been able to actually comb through it because the machines we have built are generating this information through experiments on our behalf, and we haven't even begun to analyze most of the data in a coherent way. So there is a lot of opportunity to do research without actually having to do lab work, and that's kind of fun, especially if you can't afford a lab. And it also enables people like myself and anyone else who has access to the internet to be able to make contributions to the scientific enterprise just by doing Google searches and visiting websites and knowledge bases and databases. It's a phenomenal time to be alive if you want to do science. So I do want to offer tribute to the hard work of the scientists in the laboratory and the engineers who put together these robots and these fabulous machines, and the chemists and the physicists and the computational biologists. They have found a way to do all this investigation on our behalf and make it available for free. The federal government has paid for a lot of this research. Therefore, by law, it has to be made freely available to the public. And that's what we're going to do. We're going to start mining the data that is freely available to us by federal law, because your hard-earned taxpayer dollars had paid for this research. So before I play this video, Katie, do you have any questions about what I just said? I hope that gets you a little bit excited about what we're going to try to do.
No, yeah, that makes sense. I'm excited to learn.
All right, so I'm going to share my screen. And let me make sure that I could share my audio. Also share audio system. Okay, so sometimes we don't have enough internet channel capacity to run this. It could be choppy. There could be problems when I run videos, but we'll just deal with it, okay? If we get cut off, we'll try to reconnect somehow, at least through emails. Right, Katie? Because sometimes when I run these videos, the internet goes crazy. All right, here we go. And I'm trying to get the layout here just right. Okay.
Supercoils can be positive, coiling in one direction, or negative, coiling in the other. Positive supercoiling results from overwinding, like this rope. Negative supercoiling is the opposite. If you unwind a rope and then release the tension on it, it will supercoil in the opposite direction, creating negative supercoils. Let's consider what happens as DNA unwinds during replication. As DNA unwinds, it acts like this rope when we pull apart its two strands. As you pull the strands apart, twisting tension builds up in the rest of the coiled portion. It is actually adding one twist to the remaining rope for each twist pulled out of it. At some point, you can't separate the strands anymore. The remaining rope is too tightly twisted. If you relax your tension on the rope, it will twist around itself in a supercoil. It is releasing tension. If you want to keep pulling the rope apart, you have to release the tension periodically, and one way to do this is to cut the rope and splice it back together. This problem has been best characterized in small circular DNAs. There are two methods of dealing with this problem in DNA. One cuts only one strand of the DNA double helix, and the other cuts both strands. Let's look at the first. Topoisomerase 1 enzymes cut a single strand of the double helix, pass the other strand through the cut, and reseal the break, relaxing the overwound molecule, which now has one fewer twist. Topoisomerase 2 enzymes do the same thing but with both strands of the double helix. Topoisomerase 2 cuts both strands of a double-stranded DNA and passes another double strand through the break and then reseals the break. So if a molecule of DNA is supercoiled, topoisomerase 2 can remove the supercoiling, 2 twists at a time, to yield a relaxed circle.
Is it true that medications like gabapentin could actually...
So, what do you think?
I think it's a super interesting process. I didn't realize that, like, I know that when DNA is like, it coils, like the double helix will coil, but I didn't realize like the whole thing will coil too. Like into super coil, I think is what it was saying. So.
Oh, I learned something there. I worked so much with topoisomerase 2s, I forgot about topoisomerase 1. And it did point out topoisomerase 1 only cuts one of the strands. Mm-hmm. And I don't work with topoisomerase 1 at all. I posed this question just for your information. I've posed this question to biochemists. who worked in the drug industry. I said, if you didn't know, about a topoisomerase, but if I told you, can you make a chemical? Can you make some protein from scratch that can do what topoisomerase does? And they said no. They said, and I can 100% guarantee no one on the planet could. Not your best researchers. The problem is so difficult.
Mm-hmm.
And that's one reason I just think it's really cool. If we were trying to even if you had string that was all tangled up, it'd be very difficult to make a machine that could untangle it, whatever way it would work. And this is what we have to do at the molecular level. It's just amazing. So that's why I really like it. It's just a cool protein. Do you have any questions before we move on?
I was wondering, like, how is the topoisomerase powered? Like, is it powered by like ATP or something, or is it just
Yes, it is powered by ATP. Oh, well, topoisomerase 2s are powered by ATP. The topoisomerase 1 I don't think is powered, so it just catalyzes the reaction. So it's like, where's the reaction energy? So when you start now, I don't quite know exactly how they taught general chemistry to you. Definitely in organic, definitely in certain biochem textbooks, they talk about the Gibbs free energy. Have you worked at all with that term?
It sounds familiar, but I don't remember what it is.
Okay, it's basically if you have two reactants, like for example, if you start a fire, you have the oxygen and you have the fuel, which could be wood or paper. It's energetically favorable. There's lots of energy to fuel the reaction, so to speak. And apparently there is energy available in the cell, not necessarily in the form of ATP, that can generate the cutting process. How that works, I don't know that anyone yet on the planet understands the biophysics of it. I actually am working on biophysics problems. So the cell, there is a lot of interesting energy processes in the cell. So if you've ever seen a sailing ship on television or whatever, it's just amazing that the random currents of wind, it can be leveraged to be able to get ships to sail across the sea. Well, in the cell, there's a lot of random motion. For lack of a better word, I'll call it Brownian motions. It's not technically correct, but that's a good approximation. Have you seen in any of your classes, have they covered Brownian motion? Like when you look in a microscope and you see the bacteria jumping around?
Probably. I'm not I don't recognize the term, but I've definitely looked at stuff jumping around.
Okay. Well, there's a lot of just agitation that's happening in there, and it can be leveraged in certain cases to energize a cutting reaction. I will tell you this: the biophysicists are amazed at how energy efficient processes in biology are. It's like it seems like it's using next to no energy to be able to cut and untangle. That just kind of blows my mind, So, do you have any other questions? And I'm still looking for what I had promised you, and I feel so bad because last session I actually had I had some diagrams I wanted to show you, but anyway, if you don't have any other questions, go ahead and ask me some questions while I try to look this up. If not, you might have an uncomfortable pause here while I try to figure out where stuff is.
So you said that there were multiple families of the topoisomerases. Is there do you mean there's a topoisomerase 3 and 4, or what do you mean by that?
Okay, yes, there are. So and this is where there's naming conventions have added to the confusion. So you actually have two major groups, class 1 and class 2. So the class 1 cuts only one strand, the class 2 cuts both strands.
Mhm.
But you can have, but then they started to number the topoisomerases before they started to actually affix numbers to them that had nothing to do with what class they fit in.
Oh.
So you have topoisomerase 1, topoisomerase 2, topoisomerase 3, 4, 5, and 6. And then they go into either class 1 or class 2. If that doesn't confuse you, I don't know what will. And what is what is also very, very interesting, and I know it would be an unsolved problem in evolutionary biology, and I don't think it's one they'll ever solve because it's so difficult, is in bacteria, you need two genes to make a topoisomerase because it makes it in parts.
Okay.
Whereas in humans, for topoisomerase 2 alpha, which is the one I really study, you only need one gene. And so there are all sorts of varieties, and they do different things depending on the organism you're in. Just to give you a taste for what crazy experiments we've done, I'm not saying that we should do this. I don't know that I'd have the patience. They took the topoisomerase alpha out of a mouse, and it died instantly.
Mm-hmm.
I mean, it didn't just die; it just couldn't even be born because its cells were immediately unviable.
Mm-hmm.
They took out the topoisomerase 2 beta, and the thing eventually died. After being born, it hurt the nervous system. So, if you knock out both, you'll die. So it seems like at least we found out that the topoisomerase alpha, and since you're familiar with the central dogma of molecular biology, it's very important for transcription and translation. The topoisomerase beta does some other tasks that we haven't really figured out yet. And so we transplanted the mouse. We said, okay, let's see how special these are. So we took the mouse topoisomerase alpha, put it in a yeast, and the yeast worked with it. And then we did the same with topoisomerase beta. And so it's like, obviously the yeast doesn't care that much about which one it has. However, the mouse body seems to really care because it can barely get by with topoisomerase beta. And so there's some things that topoisomerase alpha does in the mouse that beta can't do, and we haven't figured that out. And since we're similar to mice, we also have topoisomerase alpha and topoisomerase beta. So that was a really, really long answer to your question, are there varieties? And the answer is yes. Okay. So I don't know if we have topoisomerase 6 in humans. There are other creatures that have other versions of it. And as we go through UNIPROT, and just like last week, we saw the sequences of amino acids spelled out with English alphabetic letters. Do you remember that?
Yeah.
Okay. And that was the FASTA format. The spelling of the major classes are so different that one evolutionary biologist said that all these had to have independent origins. Just like, well, kind of like out of nowhere. Because there's nothing that looks like an ancestor of a topoisomerase. The reason for it, and I'm just going to point this out. Can you imagine if you had a half-formed topoisomerase? Okay, let's say you had a topoisomerase that could only cut, but it couldn't reassemble. That'd be really bad, wouldn't it?
Yeah.
Because it'd go through and shred the genome and never reconnect anything.
Mm-hmm.
It would also be really bad if it can reconnect things, but it never cuts in the first place.
Mm-hmm.
Right?
Yeah.
Instant death. So what if it could cut and it could reconnect, but it can't untangle, doing that little move where it passes, it rearranges the DNA before reconnecting? That would be bad. Or what if it can't detect the tangle in the first place? I mean, it wouldn't, if it can't detect the tangle, then it could be just sitting there doing nothing.
Mm-hmm.
So what this tells you is, how can this evolve in pieces gradually? Do you see the problem?
Yeah, I think.
Oh, great.
Yeah, it had to have been like one thing formed at the beginning, right?
Right. I call that all or nothing.
Mm-hmm.
In intelligent design literature, you'll hear the word irreducible complexity. I avoid that term.
Mm-hmm.
I mean, it's kind of the similar idea, but I'll just say, look, you have a system that's all or nothing for the critical parts. There might be a few things here you could tweak. You could definitely tweak a few things, but if you knock out its ability to reconnect, the thing is dead. And it's like, how can you evolve this gradually? So this is an example where a Darwinian process. Darwin thought you could just take slight, incremental improvements, and through the process of what he called natural selection, which is a misnomer, but we'll go with it because that term is used so much in biology, that the survival of the fittest, it'll incrementally get better. And so topoisomerase is a very good example of, that's a situation where that will definitely not work.
Mm-hmm.
So yes, we could have situations where if the creature gets a little taller or bigger or more muscular, with each generation, the Darwinian process could work there, but not for something like topoisomerase. And for that matter, many highly complex proteins, it just won't work. It's all or nothing. So I hope that was informative. So if you ever have a discussion with anyone about, does Darwinian evolution work? It's like, well, it sort of does for some things, but it'll totally fail for others. Topoisomerase is my favorite example. I personally have challenged evolutionary biologists on this, and they just pretend like it doesn't exist as a problem. And I'll say, look, whether this is about abiogenesis or organic evolution, it's a problem. You can't keep pretending like it's been solved.
Yeah.
Do you have any thoughts? And I found our paper, by the way. So if you have any thoughts.
No, we can look at the paper.
Okay. So, and this will tie to UNIPROT, and we're going to go right to the topoisomerase enzyme. Thank you, everyone, for your forbearance. We're doing this. The computer's bringing this up. Hang on. Let me. The computer's bringing this up. My computer's slow. Does Word load very slowly on your computer?
Sometimes.
Okay. It's still taking time. I'm sorry.
Okay.
Okay, so let me make sure I got actually the right document. I found the right folder. Do I have pictures of Topo? Oh, yes. Beautiful. Beautiful. Okay. So I found the paper, and just for this, for everyone's sake, I'll show it briefly on the screen, but we're going to also go to the UNIPROT website. Katie, the proper way to pronounce it's UNIPROT, as in protein, but I always call it PROT. I mean, some people might get on your case for saying PROT, so I just wanted to teach you the right way. But you'll probably catch me using the wrong pronunciation. My colleague Joe DeWeese, who is the lead author on our publications on topoisomerase, he said it's not amine, it's amine. It's like, okay. Like amino acids.
Yeah.
It's not amino acids, it's amino acids. I'm like, okay, Joe. So let's do this. By the way, was the audio adequate in the video I shared? It was good?
Mm-hmm.
Okay. So here, here is a color-coded topoisomerase FASTA sequence. So do you remember what FASTA is? It is a way of representing the amino acids using English alphabetic letters.
Like the primary structure?
Yes. It's a way of you're really sharp. Yes. It's a way of representing the primary structure. There are alternate ways of representing it, but that's one way. And while we're at it, I'm going to bring up another diagram because this, see if I could find it. I'm going to put this all in one place so we could refer to it more efficiently the next time we might encounter the need for it. Let me see if I could find this. So I had it last time. I had a PowerPoint on zinc fingers, but it actually shows the representations. And while this is coming up, again, the PowerPoint's going to take about a minute to load on my system. So in the meantime, I'm sorry to be making you wait for
It's all okay.
you know, when I make a presentation, everything's all queued up. And I remember I had to make a presentation recently. It took me a week to prepare it. It was they only gave me 13 minutes to deliver. I had everything queued up and I was talking as fast as I could. So there are times I'll be more efficient than this, but I'm really glad we could be a little bit laid back. So let me, what sort of, do you use Google or do you use Chrome?
Yeah, I use
which one? Oh, go on. I interrupted you.
Oh, no, I was just saying I use Chrome as like a internet browser or?
Yeah, internet. I have to be careful. Sometimes I get so much garbage on my, you know, there are ways to use, there's this browser called Brave. It blocks all the ads. And some of the ads I get are just like, you know, that's not really family friendly.
Mm-hmm.
I'm just mentioning it because I have to be careful since I'm making videos. Sometimes stuff will pop up that I'm just so embarrassed about. Like, oh my goodness. I don't know if do you get any of those?
Sometimes, yeah.
I just saw one and I mean, it was just for a handbag and it's still like, you know, I didn't like the way they were trying to advertise it. Mm-hmm. So sorry, I had to bring up a clean browser here. So let's go to the UNIPROT website. And there's an easy way to get there. I'll just type in UNIPROT. And it should take me right there. And then I'll just click here. And it says find your protein. Now, I'll just say this. Unfortunately, just like learning a lot of systems and working with stuff, it really helps if there's someone that actually knows, you know, has worked with this and they can teach you. Because there's some things that are not very intuitive and they're like names. Mm-hmm. They're naming conventions that I might say topoisomerase, but there's so many naming conventions to get you exactly the one that you're looking for. And unless you're in a community that studies what you're interested in, you might not have, you might be looking in the wrong spot. So if you excuse me, let me close this door here. So, because I happen, and there have been times I've been going down rabbit trails, looking at the wrong protein for like an entire week until someone pointed out, it's like, no, that's the wrong organism, the wrong protein. I was so embarrassed. So I just wanted to let you know, it does help to be integrated in a research community where you can rely on other people to give you the right names for proteins, because they're like colloquial names, and then they're the official names, and then sometimes in peer review publications you can't tell what they're talking about because it's so specialized. But anyway, thankfully, topoisomerase 2 alpha is well known. The one of the official names is top2A. Top2A.
Okay.
And so we'll just type this in, and it'll give a whole list. And so we have top2A. So top2A is topoisomerase 2 alpha. And they have human, mouse, rat. I don't know what a Krieger is. Can you let me satisfy my curiosity? I want to know what it is. A Cricatulus griseus, a Chinese hamster. Okay. Okay, this is bad. You could tell I'm just curious about everything. I would literally almost just look at all these just to find out the names of these creatures. I'm sorry. So there is what they call a session number. A session number. Like there's a unique identifier, just like a social security number. And so they have all these identifiers and stuff. So if researchers really want to be sure we're talking about the same protein, I mean, there are a bazillion of these proteins in the database. Just to be sure you're talking about the same one, they use identifiers, which is good. So I'm just going to walk through this. And please stop me if you have any questions. So I'm just going to give you a tour, and then we'll go to the paper.
Okay.
Do you have to check out at exactly 3 o'clock? I do want to respect your time.
No, I can stay longer.
Okay. So I'm just going to read this just to acquaint you with what you're going to see. And the best way sometimes to learn this is to just play with it and say, look, I probably understood only 5% of what it said on this page. But you slowly start to learn a few things, and then you talk to researchers. You go back and forth. You ping everyone you can possibly know and say, do you know anything about this? And most of the time they'll say no, but you might meet someone who will help you. So I just wanted to give you a few pointers. It's not unusual to be totally lost when you begin. So topoisomerase, so let's see what it has to say. Key decatenating enzyme. Okay, so all right, let me see what that word means. Decatenating. All right, so I literally sometimes when I've been reading a scientific paper, I'm just like, in the first 200 words, I think I probably spent 3 hours just looking up what the terms mean. And then those are explained by even other words that are just miserably difficult to understand. So before you know it, you spent 2 hours and you haven't even gotten through like the first few sentences. That's normal. Just trying to give you some encouragement here. A key decatenating enzyme that alters DNA topology by binding to 2 double-stranded DNA molecules, generating a double-stranded break in one of the strands, passing the intact strand through the broken strand, and religating the broken strand. So isn't it nice that we've actually had a video to show that description? Because if I had to read this, I don't know that I'd be able to describe it in my head what it does. So videos really, really help. And that's if you have a biochemistry teacher that uses videos and diagrams, that's going to be helpful. Eukaryotic topoisomerase 1 and 2 can relax both negative and positive supercoils, whereas prokaryotic enzymes relax only negative supercoils. Okay, that's biophysics there. I actually don't know the difference between positive and negative supercoils. Catalytic activity, ATP-dependent breakage, passage, and rejoining of double-stranded DNA. I will show you there are some videos that like my colleague Joe DeWeese did on this, where he talks about this. And they have cofactors, magnesium 2+ ion and the calcium 2+ ion. I Ca is calcium.
Mm-hmm.
So when you go into biochemistry, there has to be a lot of other chemicals present to let proteins do their things, and they sometimes call that cofactors. So that has to be somewhere in the solution.
Okay.
Activity regulation, features. Okay, we're going to go back to this. So look at this, the features, and you'll notice, okay, so how long is the topoisomerase enzyme? It is 1,531 amino acids.
Mm-hmm.
Amino acids, 1,531. And we term the locations of the amino acids in terms of what they call coordinates.
Mm-hmm.
So I can go along the, see right here, it says between 79, position 79 and 108, this is what it looks like. Proline, glycine, leucine. Y is tyrosine, lysine, isoleucine, phenylalanine, aspartic acid, glutamic acid, et cetera, et cetera. Sorry, that was just practice for me. Some of these guys can spout it off really fast. So you can see here, this is a little slider that goes across all 1,531 amino acids,
Mm-hmm.
and then you can actually see it, and you can play with this. And so you have the precise slider that can go, to very specific ones. So this is phenylalanine at position 85, and then you have this big slider that can,
Oh,
take
wow.
you all the way to the end. And let's see, there should be a stop codon at the end. Let's see if it gives you a stop. Nope, no stop codon.
Hmm.
That's the DNA side. There's not technically a stop on a protein at the carboxy terminus. All right, so just bear that in mind. And they have all these sites here that they actually identify. I mean, this is almost jaw-dropping.
Mm-hmm.
They've identified the function at these specific positions. I mean, that's just kind of mind-blowing. So
Yeah.
each of these 1,500 amino acids have function. So with a lot of agony, I'm able to diagram. So you saw all those the lists there of what this does and what that does.
Mm-hmm.
So like let's see. Um, did I lose my? So it had a site here, MG2 +1, and it has a catalytic at position 541. And so MG, if this is position 541 here, I don't know if, let me try to get this a little bigger. A magnesium will bind to, that means it's connect. We use the word bind. Will bind to that, the D, the aspartic acid here. Can you see my pointer?
Yeah.
Okay, let me try to make it a little bigger for everyone to see. The magnesium will bind there. I don't know if that's 541, but as they were describing all the sites, I literally color coded it based on that UNIPROT data.
Mm-hmm.
This is what it looks like before you make the pretty picture. Do you have any questions?
Not right now.
You can kind of guess how agonizing that would be.
Yeah.
That took me all day, and let me see if we can. And there are even more feature sites. So, but we'll just keep scrolling through this. So I actually am not familiar. They've added stuff since I've worked with this,
Mm-hmm.
but you could see the things that they study: DNA binding, transcription factor activity, RNA binding, catalytic activity, signal receptor binding, structural molecular activity. I
Mm-hmm.
mean, this is just insane what these things can do. Enzyme regulator activity. So when we have a blue thing, that means there's probably been a study. And then we have all these things that there's not been any study, or that it doesn't apply.
Mm-hmm.
So let's look here. ATP binding. I'm just going to click. Let's see what happens. Open link. I'm sorry. It says go nuts. um, Go means gene ontology. So they just
Oh.
added nuts to it. So go nuts. Gene ontology. Um, and I have to confess, I've not worked very much with my colleague Jody Weiss will understand this, but I mean, this is so overwhelming because there's just so much data, and all they can do sometimes is just report all the experiments that are involved, and then they connect you to the publication that reported on an experiment that was trying to figure out what this little amino acid did. So I'm sorry to skip because they've changed the format of how this worked, but I used to be able to go to this site and then be able to, painstakingly start to colorize the role of all the pieces.
Mm-hmm.
But it's just amazing to see what they are. Do you have any comments or questions?
Not. No, I don't think so.
Okay, so I'm just going to go through this. So we talked about the powering. So we have ATP binding, and then it has chromatin binding, DNA binding. So it's so that means it has to connect to multiple other things to do its job, which is amazing, just like a car part.
Mm-hmm.
Wow. Apoptotic chromosome condensation. Whoa. It's involved in that. It's involved in chromatin segregation, chromatin organization, topological change, embryonic cleavage, female meiotic nuclear division. I mean, topoisomerase it's not just cutting and untangling. It does all these other functions.
Oh, wow.
Yeah, this is, I mean, I may have just glossed over this. Oh, sister chromatin segregation, chromatid segregation.
Mm-hmm.
I do know that its counterpart, the topoisomerase beta, is involved in topologically associated domains, and that's another crazy, insane, ingenious feature. And yeah, you literally do go nuts. So it's kind of cute they're using the word go nuts.
Yeah.
Okay, so they have stuff on enzyme pathways. Okay, so this is definitely for professionals. Now, when I have worked with this website, sometimes I just go there to do what I need to, and I don't necessarily, I thankfully have to work with all the other pieces.
Mm-hmm.
So, and here they have the taxonomy. You can see how we're classified. We're classified as Homo,
Mm-hmm.
and we're part of great apes, and we're vertebrates and eukaryotes, and we're cellular organisms.
Yeah.
Okay. It is important that they have so much data, because when the labs do these research, the labs may not actually agree on, because a topoisomerase in one person might not be the same as in another. So they have a catalog of all these variants.
Mm-hmm.
So that's also important. So subcellular locations. So it resides in the centrosome, nucleolus, and eukaryotic organisms. I actually don't know what a P-body is.
Yeah, I was gonna ask that.
I don't know what that is. Good question. Okay, it stuck both of us.
Mm.
Let's see. Disease and variant features. Okay, see. So some people have variations in just 2 locations, and it's going to affect the activity. I mean, it may not take a lot to knock it out. Sometimes it doesn't do anything, but I mean, this is just how sensitive it is to change in some places. And what Joe DeWeese told me, he said, well, there could be other variations, but he said, sadly, those are for individuals that just didn't even get born because it was so severe that we have no record of it. We can find out when we play with mice. I'm sorry, just to take a little break here. Let me get a spec on. I almost never feel sorry for mice or rats, but when I saw the cruel experiments we were doing on them, I started to feel sorry for them. And I'm sorry, this is kind of a what I call a teachable moment. If it, for creationists, they are rightly to be a little concerned that we look very similar to chimps, and there does seem to be a progression from simple to complex all the way to humans. So if I, said, okay, here's a bacteria, here's a chimpanzee, here's a human, does that suggest a transition? I'll say, yeah, because the chimpanzee looks closer to us than the bacteria. And I said, okay, how about I give you a set of like a bacteria, a mouse, and a human? Does that suggest a transitional? It's like, well, yeah, it seems like there's a progression. So the way I've tried to explain this, I said, look, there are problems in going from definitely from prokaryote to eukaryote. There are problems going from, unicellular creature to a vertebrate mammal. There's so much machinery that has to change. Even if we assume common descent, we need miracles. But if we are all specially created, which is what I believe, a miraculous process that made all the creatures fully formed. There's scientific reasons I would believe that, because just like with the topoisomerase, it's either all there at once or it's dead. There are a lot of things where it's like it makes more sense that it's all there at once rather than evolving slowly. So why would God make chimpanzees so similar to us? And it took a while, but it was when I was studying proteins and systems in biology. I said, how'd you discover that? They said, well, we were using embryonic stem cells, and my heart sunk. I realized they'd taken this from living human beings, aborted fetuses, formerly living. They were from aborted fetuses. And I said, it's very merciful that we can actually study human biology without having to dissect each other. Mm-hmm. So, end of sermon there. I just wanted to say, as you study this, you will see that chimpanzees and mice are very, very similar to us. It's something to be extremely grateful for, because they are called model organisms. It is that we believe they're similar enough to humans that we can learn a lot about our own biology without having to sacrifice our own body parts. So, end of sermon. So we'll go back to this. If you have any questions or comments while I try to bring this back up.
No, I think what you said was true. Good. Yeah.
Okay, so let's go back to this. So there could be a lot of places where a mutation could be very deadly. We actually, sadly, may not discover it. Through humans, it might have to be done by doing that with mice. And which we did. We found out if you knock out the entire 2 alpha in a mouse, it's dead. And so sometimes they have all of these databases specific organisms. Are you familiar with post-translational modifications?
That sounds familiar. What is that again?
Okay. So let me this is what's beautiful about graphics. I can illustrate. So
Or wait, is that like splicing or is that something different?
it's different.
Okay.
So this is where it gets really insane. I mean, you just look at the regions of the topoisomerase here, and you're like, wow, it has all these functions. I do know, like, if you knock out the D here,
Mm-hmm.
topoisomerase is disabled. It's that it's that critical. They're let me see, the ATPase binding site. Okay, so these bright red areas are where ATP. That's where the ATP parks. That's kind of the where the gas station connects, or that's where the electric plug would connect, so to speak, to give it energy. So if you knock this out, the topoisomerase is gone too in those regions. And there are other critical regions. Yeah. So what happens is, so this is a different rendering of the same topoisomerase protein, 1,531 amino acids. And the things that are colored are where there is a chemical modification to the amino acid. So let me see if I could bring, I'm gonna bring up a, see if I could bring up a picture of post-translational modification. And I'll tell you an interesting story. John Sanford, he was my boss for 7 years. He focused so much on genetics, and when I showed him post-translational modifications, his jaw dropped. So let me see if I could find a nice diagram of a post-translational modification. You can look it up on Wikipedia, but I'd rather be able to like show you a picture. See if I could find a nice picture that will show a post. Oh, here's a good one. All right, let me re-enable this screen share. Okay, so this, I'm going to get this wrong. Carboxylation. Okay, this is the N-terminus. So when we read the proteins from left to right, so like when I go here, the start of the protein is what they call the N-terminus. Usually it begins with an M, but not always. And the end of the protein is the C-terminus, or carboxy or carboxyl, various names for it, terminus. That's just like the end of the protein. So we think of this as the beginning and the end. But, to be very precise, there's certain chemical markers that will say beginning and end. So that's how they, the beginning is the N term. N is for like the amino terminus. That's amino functional group in organic chemistry. And then the carboxy is a carboxy functional group in organic chemistry. So N terminus is beginning, C terminus is end. So when I was showing this picture, so this is the beginning of the protein. And somewhere you could get a serine amino acid, and you can actually add a phosphorus group to it. This is like, have you ever decorated a Christmas tree?
Yeah.
Right, you have these ornaments, right? So look at these post-translational modifications as ornaments.
Okay.
And they are very critical to the functioning of the protein. There has to be special machinery to locate the right location in the topoisomerase and then add the ornament. You have a special machine just to add ornaments.
Wow.
Yeah, isn't that cool? And also structurally, you'll hear this term, they will sometimes add ornaments like a sulfur group to a cysteine and then another, and that will create a disulfide bridge. And that's very important to help fold it and make it stable.
Mm-hmm.
Now, these have all sorts of chemical properties, but they can also act like signals. So you can actually modify this, and you can change the state of the protein. It actually will convey information to the rest of the cell. And we're only learning about some of this. Where it's really prominent is in chromatin modifications, which is another topic. But what I did is I looked at all the post-translational modifications, and they're colorized here. Can you see them?
Mm-hmm.
So when I use purple, purple means it's a phosphorylation.
Okay.
So let's look at, see, a serine is phosphorylated, and S represents serine. So look at all the places that the serines are phosphorylated.
Mm-hmm.
Threonines, the T, can also be phosphorylated. Now, what should be interesting is there's clearly some serines that are not.
Mm-hmm.
I mean, there's so many of them. There's a serine there, a serine there. How come this one gets it and the others don't?
Yeah.
And there needs to be, can you imagine how much cellular machinery there has to be able to be sure that you only flick certain ones and not others?
Mm-hmm.
And it also depends on the cell type and the cell phase. This is just like mind-boggling how complex this is.
Yeah.
Do you have any thoughts? I mean, to me, this just makes me like go, wow.
Yeah, I mean, it just sounds like such a particular process. I feel like, yeah, I definitely want to know like why it's some serines instead of all of them. And yeah, it sounds really interesting though.
Yes, and for what it's worth, we still don't know some of it. So when I talked about, remember I said we could throw this in a yeast, and if it's the mouse version, the yeast will be fine.
Mm-hmm.
So it doesn't, for yeast, it doesn't seem to care about all these post-translational modifications. It doesn't care. You know, I mean, it'd be a little difficult explanation as to why, you know, but it doesn't care. However, obviously the mouse cares, because
Yeah.
if I start playing with this, the poor little mouse will not be a mouse. So, and we have here, I forgot what the reds were. The blue is a methylation, and I forgot what the K's are. Sumylation. So let's look at some of the kinds. So you have OH, which is a hydroxylation.
Mm-hmm.
We have methylation. We have disulfide cross-linking, carboxylation. But there are even some more like assimilation. I mean, there are just so many of them. This is just an approximation.
Mm-hmm.
In fact, there are so many. Let me see. I wish I didn't kill that diagram. That was such a nice diagram. Let's see. Okay, I still have it. I'm going to show you another one here that just lists. Let's see. Oh, that's a terrible diagram. Let me try to get another one. I'm sorry. Let me type through this. This is see if I could get one that gives a nice long list of I'll just show this one, and then we'll move on. We don't need to belabor it at this stage. So this is a different protein called a histone, but the histones have a lot of post-translational modifications.
Mm-hmm.
So Me is methylation, Ci is citrulation, we have acetylation, we have ubiquination, phosphorylation, which we had already talked about. Then there's sumoylations. There are just so many. And Dr. Sanford saw this, his jaw dropped. He said, I didn't realize that they were just that complex.
Mm-hmm.
So I guess we'll just move on here. And so let's see. Where did I put? There it is. So that's post-translational modification. Oh, okay. So you see here how it's giving you a position, like 17, 83, 101, 131.
Mm-hmm.
Can you guess how I figured out where to put the red and purple marks?
I don't know. Did you count?
This is where it was agony. I had to figure out a few tricks. So do I have to go like, okay, do I go up to number 83 here? So 1, 2, 3, 4, 5. I didn't do it that bad, but it was close. Okay.
Okay.
If I could get to the right region and then I'd use the search tools, I could identify it, but it would be an absolute pain if I did it that way. Right.
Yeah.
And so I would color code this. So why am I showing you this? This is where there could be, no one has done this sort of work of visualizing it.
Mm-hmm.
This would be a very nice project to just say, hey, look, I want to present this as a project. They're probably efficient way, if there's someone who can do computer code, they could probably do this even faster than I did it here.
Mm-hmm.
But you could see this is a day-long project to be able to take the UNIPROT coordinates
Mm-hmm.
and then start to colorize it. So let me see. Let me see if I can get this a little smaller. I'm trying to find. So position 1 is a modified residue, cross-link. Okay, so the Ks, it used to give me the data on what kind of sumoylated lysine. Sumoylated. Sumo is ubiquitin-like something. I don't know.
Mm-hmm.
So anyway, so let's just see some of the coordinates and then we'll move on. I'm sorry to be, this can be very tedious, but this is science is often very tedious.
Mm-hmm.
And there's you'll be throwing a lot of facts that don't seem to be connected. And you're very lucky when you can kind of connect the dots. It's like a big puzzle. Have you ever put together a jigsaw puzzle?
Yeah.
Yeah, we're trying to put together a jigsaw puzzle. Sometimes will not even get anywhere near the full picture, but you get a little bit of it. And biology is so complicated, we'll never get the whole big picture.
Mm-hmm.
So, okay, so I have on here coordinate 1387. And mercifully, it will give you the letters to the left and to the right.
Mm-hmm.
So ADDVKGS. Let me type this in to okay, I'll show you my little trick because you might have to this might come in handy for some of the stuff you might want to do. Mm-hmm. So I'm just going to type this in. ADDVKGSVPLS. Okay, so now I can, maybe if I'm lucky, I can do a text search.
Mm-hmm.
Let's see. Will it even let me do a text search here? Oh, I'm not enabled for a text search, but I can't seem to do a text search. Well, I have another, what do you call it? I have another word processor
Mm-hmm.
where I could put that in and then I could just search that string. So let me cheat a little bit here and just show you how I would do it. Oh, I can't even copy it. So let me grab it from UNIPROT. 1 second. Do you have any questions while I'm trying to get through all of this?
No.
No questions. Okay. So let's try that again. Okay. Do you remember the name of the official name of the protein? Top.
Top two, yeah.
You're gonna learn this like the back of your hand. All right. So let's go to the gene sequence and we'll download it. Now I could do a text search. And that was what? ADDVKGS VLPS. See if we'll find it for me. Okay. Now sometimes the wraparound will make it difficult for you to find. So let me just shorten it. Nope, didn't find it. Let me just look for S.
Is it the plus sign?
That might be it. Okay. There it is. ADDVKGS. So if I were looking, you got it. Thank you. Well done, researcher. So when we go to here, I don't even remember where this was. It was just some random serine, but you could see now I didn't have to actually physically count every position.
Yeah.
And sometimes it's like, okay, see, I mean, there are little tricks you can do, and you do your best. You hope you don't make a mistake. Do you want to just pick a number here, and we'll just do this exercise one more time so we can just see how it's done?
You can do 954.
Okay, let's see if we have one there. I don't have that number.
Okay, just do 949.
Okay, it's assimilated lysine. So then you click on here, and it's very nice. It's going to give you, I don't know if I can, nope, I can't. So 949, but I can go here and say, okay, let's look at the string. And you could just go a little bit left or right. So NGTEKTPP. And we're looking for the K in the center. So now let's see if we can find the needle in the haystack. Okay, using a search function. There it is. So now I could say that K and G, and then I'll find it somewhere here. Do you see the process? Does that look painful?
Not too bad.
It's not too bad. It's more tedious than thinking, okay? But the net result is you come up with something that's very, I mean, John Sanford was my boss. He had been a geneticist for 40 years. And when he saw this, his jaw dropped. He said, that's amazing, Sal. And it wasn't like, I said, look, I can't take credit for, this is already so well known within our little, you know, kind of our little bubble of protein biology. But this is where this is where we have a situation where in one group, this is common knowledge, and in a group that's also very respected, the geneticists, they had no idea.
Mhm.
And so this is where I was saying that there's a lot of opportunity to do a lot of cross-disciplinary education.
Mhm.
Even for people that are senior scientists, very accomplished, there might be stuff that should be of interest to them that you have to bring to their attention that they're not aware of.
Mhm.
And I've taught, even a variety of biologists. I'll say, did you know about that? And it's like, I sort of studied that. And then but you show them a picture, it's like, wow, I didn't know it was that complicated. So that's why I was thinking this is going to be awesome stuff to get involved in, because there's definitely a need to be able to act as a go-between, a liaison between separate disciplines.
Mhm.
So, okay, okay, we're 3:20. Okay, I'll try to just, if we're not able to finish all this, but I showed you kind of the techniques to be able to see. A lot of what I just did for a paper that is not yet published was just doing what you just saw. We went through a knowledge base, and we're saying, hey, look, we wanted to present on the complexity of the topoisomerase protein. We think this would be very informative to people. They need to understand just how complex this is, how difficult it is to evolve, especially by our particular twist on this. We were saying, look, you cannot, there's a particular hypothesis in evolutionary theory called frame shifts. And we were saying, look, it's not going to work on something as complex as this. You're going to just blow the protein apart if you try to do a frame shift mutation. Are you familiar with frame shift mutations?
Yeah, I've heard of those.
Okay. Yeah, it would blow this thing apart because all the post-translational modifications just be blown out of the water. The spelling of the protein would be it'd become a total disaster. But there are theories in evolutionary biology that said that this could be a key evolutionary mechanism. I'm like, no, it's not for anything as complex as this.
Mhm.
So as you study and start to catalog the parts, and I'm sorry I lost it. In one of these diagrams, I actually had the nuclear localization signal identified. And so because we talked about nuclear localization, let's see if we can find anything with localization. So I'm just going to go to the top here and see if it has the word localization. Localization. I don't know how I found it. I might have had to go through another paper to find the localization, but you can find what the localization string is.
Mhm.
And that was something I had in mind that I'd like to find what the nuclear localization signal is in topoisomerase 2 alpha. I mean, I found it before, but I don't know how to replicate finding it again.
Mm.
So, oh my goodness, protein interaction databases. Topoisomerase 2 alpha is indicated to interact with like 100 different proteins. That means there have to be, when we look at this diagram here, there are regions that other proteins will dock with. And we know that it's going to interact with ATP in this region, in this one. But like, I've identified a few other like amino acids, other residues where it specifically will dock with its complementary copy. And look at all those positions. And that's just with one other protein, another topoisomerase. But what about other proteins? You would have diagrams like this for every protein interaction.
Mhm.
And we don't yet have all the capability to estimate those positions. This is just insane. And there are 100 different proteins that can interact. So each one will have a different like signature of where it lands. It's like different footprints. Maybe that's the way to say it, or fingerprints. Everyone will have a different fingerprint, but it has to match.
Oh, sorry. I
Okay.
think my fire alarm just went off, or the smoke alarm in my house.
Are you all right?
Yeah, I think so.
You want to check?
Oh, it stopped.
Okay.
Okay, that was kind of weird. Sorry.
Oh, maybe we're heading toward the buzzer here. So there are all these signatures or fingerprints. Have you used fingerprint identification to access any, like some smartphones will say it'll be facial recognition or fingerprints, so you don't have to type in your password? Well, so basically the interacting partners for topoisomerase would have almost the equivalent of passwords or facial recognition. It has to be able to recognize. This is just one recognition system.
Yeah.
It just makes my jaw drop that it could do this. So, I mean, the complexity is jaw dropping, but like I said, how did I learn some of this stuff? The hard way. I started at a website just like this and started to self-teach. That's I mean, you can imagine, like, I started with like knowing nothing, and I'd just look through this and I'd say, I have no clue what this means. Let me Google. And then thankfully I had a mentor in Joe DeWeese who has studied topoisomerases for like 20, 25 years. And I had a resource to talk to. I said, Joe, I have no clue what this means. Can you help me? And then I was taking classes at the NIH. So I think if anything else, when you start studying organic chemistry and biochemistry, you might actually see how it might be useful to understanding how protein systems work. So I'm going to have to go through this myself to find out, how was I able to find the nuclear localization signal? I don't know how it was able to find it. Let me try it one more time, because I think that would be a really cool thing to get involved in. Nope, it has nothing here. So they didn't load it on this database. So I think I might have had to use Google and say topoisomerase nuclear localization signal. They would give me the sequence, and then I would have to do that little trick that we had where, okay, I have the sequence. Now I'm just going to do a search, and then I can identify where it's located. So I used to think it was here. So let's just kind of wrap it up. Do you have any questions, comments, thoughts?
I mean, once again, I think it's such a interesting and complex process, very particular. So it's interesting to learn about, and I'm excited to learn more about it.
Okay, so now that you've gone through that, if you ever, put on your resume, and always be honest, that you've worked with UNIPROT, you could say, okay, I had passing exposure to the UNIPROT website. I've done some exercises where I looked at the post-translational modification of these systems and what specific, and you can identify the coordinates, serine, whatever number, and you'll be respected. At least, you've started to know your way around the city, so to speak, a little bit, because it's all here in UNIPROT. Every protein is so specialized that you can be talking about a specific protein and its post-translational modifications, and everyone else will not understand you, even professionals in the field, unless that happens to be their protein. But what that means is you can at least, even with a little bit of exposure, something that they don't. You have a skill set. You're representing that you have skill sets that are specialized that other people don't have. And even if it may not be immediately needful for that particular job, it shows that you have kind of the detective skills to be able to dig stuff up.
Yeah.
So a lot of my research for 7 years is really just using Google search engines and engines like UNIPROT, and just like trying to put together a jigsaw puzzle. You know, if you're able to connect a few pieces, it's like, yeah, you've advanced scientific knowledge a little bit, and it's just like one little paper at a time, and you know, after decades, you actually, you know, actually something collectively is advanced, but that's just the way it is. So um, I hope you know, we could do a little work with this, and actually try to glean data that even I've not explored,
Mhm.
and we can report on it. And I'll show you the process of how we might find a jigsaw puzzle piece. And what can be very frustrating is just like when you're making a jigsaw puzzle and putting all the pieces, it's like you'll grab this piece and this piece, and you don't know if they're going to connect. Most likely they won't. And you just go grab another piece just randomly.
Mhm.
Sometimes you get lucky enough to make something insightful. I don't know if that could be very discouraging to you, but since you've done jigsaw puzzles, maybe that'll put you in the right mindset.
Yeah.
I mean, when I've looked at how they discovered things, my jaw just drops. It's like you guys were just kind of almost fishing, and you got really lucky. And that's, you know, they say science is, you know, the experimental method, and you observe, you make hypotheses, but a lot of professional scientists will say, well, that's only like half the story. So much of it is luck, where you
Mhm.
just happen to be discovering something that you weren't even looking for, but then it ends up being the most important thing you probably discovered your whole life. It was something you weren't looking for.
Mhm.
And so, uh, you know, in the age of Google, we report stuff if it's published and Google search engine can find it. If someone else is trying to connect pieces of the jigsaw puzzle, he might find our work somewhere, and he'll
Mhm.
be very grateful we did that.
Yeah.
And that's kind of how I look at the current scientific enterprise as far as biology is concerned. There's so many disconnected pieces, and we're just trying to, you know, see if there's stuff we can connect. And it's amazing. Um, I'll give you the one of the best examples I know of some guy who was connecting a puzzle. It took him 20 years
Mm.
to connect the. When you take biochemistry, you'll hear about the TCA or the Krebs cycle or the citric acid cycle. It took him 20 years of very difficult going to the lab every day for 20 years. He won the Nobel Prize for figuring it out, but
Wow.
man, that was miserable. So when you look at metabolic pathways, you're like, well, that's cool. And just like, well, if it helps to appreciate how hard it was to discover that.
Mhm.
So we're part of that enterprise to discover the pieces in biology. So I sometimes go on these tangents, partly because um, unless you have a certain temperament, which I don't, I certainly need a lot of encouragement to do work like this, because it can feel like sometimes we're going at a dead end. And a lot of times we are. And there's really nothing we can do. We just have to do our job, do our best. And at least if we discover a dead end, we're basically signaling to other people. It's like, maybe look elsewhere if you want to find some treasure. So I'll meet you backstage. Do you have any closing thoughts? I'll give you the last word.
Yeah, I think. Oh, I have to put my thoughts together. um, yeah, like you were saying, it's very interesting processes, but I also know that like people aren't just like discovering these things out of luck. I feel like it's also God's hand in the matter, and like God wanting people to discover these things. So
Well, wonderful. Okay, to my audience out there who've tuned into this discussion, thank you for watching. I hope you've been blessed. You get to see the life of what scientific work can be in this particular field of biology. So until next time, take care and God bless you.